Good afternoon. My name is Scott Swing, and my supervisor for my review was Jalal Jazari. As you can see, my topic for this afternoon's presentation is non-viral gene therapy, and my belief that it is the future of medicine. So to begin, I guess the question is, well, what is gene therapy? And in a simple answer, gene therapy is the expression of genetic material as a pharmaceutical agent to treat the diseases of the human body. Now, the most common form of gene therapy is the use of recombinant DNA, which expresses and encodes for a therapeutic gene to replace a mutated gene, which leads to a disorder or disease. Another common practice is the use of DNA which codes for a protein. The expression of this protein then upregulates or downregulates specific processes in the human body. Now, the theory of gene therapy isn't a new one. Friedman and Richards published a paper in 1972 in Science called Gene Therapy for Human Genetic Diseases. Now, they thought that although hypothetically the idea of expressing genes to correct errors in genetic codes in humans which lead to diseases was possible, they believed that it should not be done in humans for specific reasons. Firstly, they believed that our understanding of the basic processes such as gene regulation and genetic recombination were simply far too inadequate. They also stated that the relationship between molecular deficiencies and the diseases is still far too rudimentary. And lastly, they went on to state that the idea and the theory of the short-term and long-term side effects were still far too unknown, and that it was still far too risky to use gene therapy. Now, we've come a long way since 1972, and our understanding of these processes is such that gene therapy is now well worth investigating. So what is the process of gene therapy? Well, basically you package your DNA into a vector, and then you insert it into the human body, usually intravenously. And then once inside the human body, the human cell machinery expresses the gene, producing the therapeutic product. Now you can deliver nucleic acids to increase the expression of a target protein, or deliver small interfering RNA, siRNA, and oligonucleotides to decrease the production of a specific protein. Now this sounds simple enough, but the reality is that the process is met with many intracellular and extracellular barriers. Both naked or encapsulated, they face physiological barriers such as the cytoplasm, blood serum, and the nucleus. A study in mice in 2002 by Harada showed that naked plasmid DNA, after being intravenously injected into the mice, was entirely degraded in just under 5 minutes. Some of the other barriers that are faced by gene therapy include DNA nucleases, which degrade the genetic material. The internalization into endosomes takes place by the vectors quite often, which then leads to lysosomes and the degradation of your material in the vector. The delivery to the specific nucleus of interest as well is an extremely complicated process. And once it is at the nucleus, the actual entry into the nucleus is a massive barrier as well. Also, exogenous DNA and RNA trigger an immense immune response, leading to a cascade of inflammatory cytokines. So how do we overcome these barriers? Well, there's several different processes that take place. One such of these is the specific modification of the oligonucleotide backbone. Now this has greatly increased the chemical stability of the nucleic acid once it's delivered. Once inside the cytosol, viruses travel along microtubules to improve endosomal escape. Non-viral particles, however, benefit from the use of endosenolytics, agents such as chloroquine. And interestingly, chloroquine, when used as this agent, is derived from bee venom. Better recognition by the cell membrane bound receptor proteins is achieved by conjugation of moids to the vectors. And this is a process called active targeting. And this active targeting is one of the largest challenges faced as far as intracellular barriers are concerned. The toxicity and immune response can be reduced by carefully tailoring the vector, as well as using the lowest effective dose. So instead of injecting the patient with a larger dose, you inject the exact amount required to induce the response in the human body. So I said that you would take your DNA and you would package it and then inject it. One of the most beneficial methods of doing this is through the use of a vector. Now, the vector is the vehicle which delivers the foreign genetic material into the cells. And this vector can either be viral or non-viral in nature. Non-viral vectors take advantage of the virus's ability to integrate into the host cell. And examples of these are the family of retroviruses, adenoviruses, and the herpes simplex. They have, however, been faced with fundamental issues, including their toxicity, their immunogenicity, and limitations when dealing with scale-up production. On September 17, 1999, 4 days after receiving an intrahepatic injection of adenovirus particles for the treatment of OTCD, 18-year-old Jesse Geslinger from Tucson, Arizona, died suffering from a massive immune response triggered by the viral vectors. This is also the first time anyone had ever died from gene therapy. Now, this led scientists and researchers to believe that viral vectors, although they are beneficial, we could potentially use the theory of how the virus infects the human and implement it into a non-viral vector. Examples of these are polymers, dendrimers, cationic lipids, and peptides, and all have potential routes for compacting DNA and delivering it. Unlike viral vectors, which have evolved mechanisms for overcoming the barriers intracellularly and extracellularly, non-viral vectors have unfortunately expressed reduced transfection efficiencies. They do, however, benefit from having low immunogenicity, as well as the ability for large upscale production. Several non-viral vectors, which are being utilized for the delivery of genetic material into the host recipients, include the lipid-based vectors. The mechanism for gene transfer of cationic lipoplexes is produced primarily through endocytosis. Once the complex has been taken up by the cell, the lipoplex destabilizes the endosomal membrane, causing a reorganization of the phospholipids, which then diffuses into the lipoplex, interacting with the cationic lipids, and causes the DNA to dissociate into the cytoplasm. Polypeptide vectors utilize short sequences of basic amino acid residues which are able to cross the plasma membrane. These peptide vectors are often used in the delivery of oligonucleotides into the cells of the human body. Polyamidamine, PAMAM, is the dendrimers, which is most widely used dendrimer-based vector for gene transfer. This is because of their commercial availability and the ease of synthesis. Lastly are the nanoparticles. Cationic carriers are a widely used form of nanoparticle vectors because of their ability to effectively condense DNA and interact with target cells. DNA polymer complexes involving cationic polymers are able to transport genes into the target cells, although the mechanism for this is still somewhat not understood. So by 2013, more than 1,800 gene therapy trials have been approved, are ongoing, or have been completed. Now this data here, saying 39 for 2012, is a little askew because the data takes a little while to get in. So where are these taking place? Thus far, 31 countries are involved in these clinical trials in gene therapy, with 65% of them taking place in the Americas, approximately 1,174 of these. Interestingly as well, Australia is noted as being 1.6% with approximately 30 trials ongoing. And this is interesting too because that number has actually increased quite significantly. From 17 in 2011 to 30 in 2013. So what are we trying to target with these? Well, as of right now, 64.4% of all of the clinical trials ongoing in gene therapy are targeting specifically cancers. Now, a lot of these are targeting the p53 tumor suppressing gene. So in conclusion, the overall trajectory of gene therapy is positive. And the increase in evidence supporting the clinical benefits in a myriad of different diseases indicates research is headed in the right direction, with non-viral gene therapy leading the way. And it's my belief that it will change the face of medicine as we know it. Thank you.